Hello and welcome to this presentation on the next generation of fluorescent proteins from the corals of Sabah. This is a recap of the presentation which was delivered at the conference organized by the Sabah Biodiversity Center entitled Triggering Biotechnology Industry in Sabah on the 25th of January 2018. This project focused on the discovery of fluorescent proteins from the corals of Sabah. We attempted to elucidate the fluorescent proteins and establish a database for future reference. Basically, corals are developed by reef-building organisms which belong to the class Anthozoa and the phylum Cnidaria. These tiny zooplankton are responsible for the development of reefs, or what are known as coral reefs. Among the corals of Sabah, we have both soft and hard corals. This is an example of a soft coral which is named Heliofungia actiniformis. As you can see, this coral exhibits both red and green fluorescence. This map gives an indication of the dive sites at which the corals were discovered. So we have listed the locations of the dive sites without indicating the exact coordinates. Permission to collect specimens was obtained from the Sabah Parks Authority in accordance with approved protocols to prevent damage to the original specimens as well as to the coral reef beds. This image shows how the corals are being acquired by our research assistant. This is documentation of the process of cataloging of the coral specimens and their storage in ice for shipment to the laboratory for subsequent DNA extraction. These are some of the images which we retrieved from the coral specimens. The corals were exposed or irradiated with blue light, which excites the green fluorescent protein, and the resultant image was captured through a yellow filter. So what you see is the fluorescence against the dark background. So this is an example of green fluorescence in Colostria species. This is another bright fluorescent signature of Triogyra sinuosa. This is under fluorescent example, Montastriata species. This is Galaxia species, which is a hard coral. As you can see, this specimen is encrusted with barnacles. This is Favete species, which shows an intense green fluorescence. This is Colostria species, which shows a fluorescence which is more in the range of cyan. So as you can see, the visual image without any kind of excitation by blue light does show some kind of fluorescence. This is an example of a coral which exhibits both red and green fluorescence. This is Mycelium elephantosus. So in the image which is irradiated with normal light, you can see some indications of red fluorescence. However, the green and red fluorescence become more intense when subject to irradiation with specific wavelengths of blue and green light. This is Euophilia ancora, or the Inca coral, which exhibits both red and green fluorescence. What needs to be noted in this case is that some of the red fluorescence may be the result of the peridinin, which is derived from the dinoflagellates that are in symbiotic association with corals. This is Cyclosporus species, which exhibits both red and green fluorescence. This is Colimia species, which is a species of button coral. As you can see, excitation with the blue wavelength results in a green as well as an orange fluorescence and the requisite red fluorescence as well. These are the Acropora. So Acropora are staghorn corals. So we examine a diverse range of Acropora. They have different morphology. And based on the results from DNA barcoding, they are actually different species. These results were published in Zootaxa, in which 12 new species of Acropora were reported from Sabah. So at the end of this study, we have developed a complete database of more than 100 specimens, which has been submitted to Sabah Biodiversity Center and will serve as a baseline for future reference. This is an overview of the current research on the project, which is bioprospecting for fluorescent proteins from corals of Sabah. Thank you for watching.